So a primary goal of our project is to generate a useful database for full development. We're both zebrafish, and eventually to have information about conditions that affect creating pictures. And we've used two largely different approaches in our groups to generate this data. In my laboratory, we're using confocal microscopy using transgenic lines that express fluorescent proteins in different cell types. And the two that have proved the most generally useful for us to generate anatomical data are pictured here, and they express green fluorescent protein in cartilage and in cherry and osteoblasts. And these are great lines for imaging. They're extremely bright. They have vertical background, and they maintain expression into adulthood. So as long as we can get light to penetrate the samples, we can see them. And this is an example of some of the data that we've generated using these transgenic lines for some wild type fish. And I would urge you to go this afternoon to Alexandra Scully's poster. She will show you many more examples of beautiful imaging data, and also talk to you about the challenges and approaches that we have in imaging with live fish. And this is one of the primary strengths of our approach, because we're looking at live animals. We can dynamically image processes during development of the skull. And so this is images of a single live fish over a 2-week period, looking at all of the cartilage and bone, dorsal part of the skull. Now, yes. Okay, so that means I have to stand back here. Okay. Yeah. Okay. All right. So now, because we're imaging live animals, that's also one of the drawbacks of our approach, because we are limited in the amount of time that we can keep individual fish on the microscope stage, and the number of times that we can iteratively look at an individual. And also, as the fish gets larger, because it's confocal microscopy, it's more difficult for the light to penetrate, and eventually we have trouble seeing all of the structures that we would like to see. Now, in the Harris lab, they use high-resolution microcomputed tomography, micro CT, and this is a nice complement to the data that we generate, because they of course, are imaging fixed samples, and they can get extremely high resolution data that's equal resolution in all axes, unlike the confocal microscopy. And so it lends itself extremely well to doing three-dimensional reconstructions. Now, both of our groups in the past few months have really focused on trying to get complete, comprehensive data sets of wild type fish on the FaceBase hub and available. And as you saw some really nice examples of that in the presentation from Carl a few minutes ago. But we have multiple scans of both male, micro CT scans of both male and female fish available, as well as well over 100 separate data sets of confocal microscopy. And all of these data are available for download, and people can take them and do what they want with them. And one thing that I want to point out about our confocal data sets, we focused on providing fairly complete sets of scans at different time points of the same individual fish. So, a user can go onto the website and basically download this imaging data over, a period of a couple weeks or longer for a single individual. Now, more recently, as you also heard Carl and Rob talk about, Osman in my lab has been taking some of our confocal data and turning it into three-dimensional surface models. Now, we actually have been creating 3D PDFs, and these have, I think, in general, out in the field, other people doing anatomical work of this type have proven to be very useful. They're relatively modest size files that can be viewed easily using, the free program Acrobat Reader. The images can be freely rotated. They also have associated interactive buttons that you can use. Both, we can label all the structures and color code them, and then you can use the buttons to make things either become translucent or take them out of the picture completely. And so once you've downloaded this file, you can look at it and play with it as much as you want on your own local computer. Now, the intention is to make all these PDFs available for download on the website. Now, unfortunately, the integrated 3D viewer is not compatible with PDFs. But as you saw, we have made a workaround so they can take the data on the hub and basically do the same thing with it. As I said, the lines that we've been using initially for most of our imaging, the transgenic lines, are basically just broadly expressed in all cartilage and all bone cells. So they're very useful for anatomical information, but they don't really tell you anything that interesting about gene expression. So we're now starting to image some additional transgenic lines that give us information about developmentally interesting gene expression patterns. So this is one nice example. We've just started imaging and trying to accumulate the data for this. So this is so the BMPER gene encodes an extracellular regulator of the BMP signaling pathway. And we actually set out to look at its regulation a couple years ago because its ortholog in zebrafish has extremely focused expression pattern in early osteoblasts in the larvae, but we really didn't know much about its expression later during skull development. And so this transgenic line is actually an enhancer that's upstream of the human BMPER gene, hooked up to GFP and driving expression in zebrafish. And Ali has more pictures on her poster you can look at, but as you can see in this example of the frontal bone, it is expressed in a subset of the osteoblasts only at the edges of bones during this period of rapid growth, and then eventually becomes downregulated as the bones approach and overlap to form sutures. So one of the goals that we talked about at the last meeting was trying to figure out a way to integrate the micro-CT data and the confocal data into a single model. And there's a lot of challenges inherent in that approach. I mean, one is that, of course, the data is generated in two completely different ways and has some different characters. But also, because the micro-CTs are based on the mineralization of the bone, it's very difficult to get good data on younger fish. And of course, for the confocal data, the younger fish are precisely what we can get the better data on. So there was sort of a limited period of overlap where we could really acquire data on the same individual. So in the Harris lab, they've developed a contrast enhancement method for the micro-CT based on silver nitrate staining. And in whole mount, the silver nitrate is as sensitive as alizarin red staining, as you can see in this quite young fish that they've stained here. And the enhanced contrast allows really beautiful imaging of much smaller samples than previously could be done. And even at later stages where it's possible to do an unenhanced micro CT, you get nicer looking data and better resolution of the structures using the silver nitrate enhancement. So this, I think, is going to go a long way towards overcoming one of those limitations. Now, we, as sort of a first step towards doing this, we decided to try this on Chihuahua mutants and some siblings. And this is an interesting mutation for a couple reasons. First, it's a mutation in one of the type 1 collagen genes. It's an accurate model for human osteogenesis imperfecta. So, not only is it a human disease model, but it's also a disease that affects matrix deposition and mineralization process of the bone. So we thought that could be a particularly interesting comparison between the gene expression from the confocal and the bone deposition that you see by micro CT. So we're still kind of at the early stages in this, but we've taken the same, in a set of fish, done the complete confocal imaging on the live fish, and then at the end of that experiment, fixed the samples and sent them to the Harris lab for micro CT scanning. So this is our first attempt at the data integration. And again, Osman at his poster can tell you more technical details about how he's doing this and the landmarks he's using to register the images. But we're pretty encouraged by these first attempts. And I think we've really made a lot of progress in overcoming the issues of the two different types of data and some of their inherent incompatibilities. So over the next few months, we're going to be doing some more of this. We're going to be using some additional mutations that we have in both of our labs that also affect matrix deposition and mineralization. And also now we can, so that first set of data was done without the silver nitrate contrast enhancement. So now that that technique is working really well, we're going to be able to do some earlier time points as well. So, we, in addition to generating the wild type data, we do want to put an emphasis on, trying to get as much data on mutants as possible up on the website, and especially focusing on things that are relevant to human disease. So this is just one example of several mutations that affect different aspects of the FGF signaling pathway, which of course is critical in human craniofacial development. And these are mutants that were either identified in forward genetic screens or generated by direct targeted mutagenesis in the Harris lab. And so this is a likely null mutation in the FGFR3 gene that has, as you can see, sort of mispatterned extra sutures. This is a knockout of just the tyrosine kinase domain of the FGFR3 gene. And this is a hypomorphic allele of the FGFR1A gene. And so they all show very distinct craniofacial phenotypes, and data on all of these will be available on the hub. And we have other mutants, including this one. This is again found in a forward genetic screen in the Harris lab. It is a still as yet unknown gene, but it is obviously very interesting in terms of human disease. It shows craniosynostosis, as you can see, fusion of the coronal suture on one side. And finally, we all have expressed a heartfelt desire to improve interactions with other spoke projects. And so one of the great things about doing these micro CT scans is they're relatively quick, technically simple. They don't require any complicated sample preparation. So they're a good screening tool for looking at fish that may perhaps be normal. And so we've been interacting with the other spoke here in Boston, headed up by Dick Moss and Eric Liao. And Eric had a very nice paper published recently looking at several mutations in the Wnt signaling pathway that affect palate formation. And he was convinced that the heterozygous carriers were completely normal and were healthy and swimming around in the tank. But we managed to persuade him to give us some for micro CT scanning. And as you might be able to appreciate from these scans, all three of these show fairly distinct craniofacial defects that would otherwise have been pretty hard to appreciate. So we're hoping that these kinds of interactions will continue in the future, and we can follow up on some of these, with more intensive studies, including crossing these mutations into transgenic lines to do confocal imaging. So just to sum up, over the next funding period, we want to continue to work on better integration of the micro CT and the confocal data, including some new mutations and different stages. We're working towards having a completely segmented three-dimensional model of the adult micro CT data, which is a bit more complicated and more challenging than the confocal, but we're getting there. And eventually we'd like to have that available on the hub as well. We're generating more confocal data on additional transgenic lines. And of course, we are always anxious to add both confocal and micro CT data on additional mutants from our labs, from other spoke projects, or eventually from other labs outside of FaceBase. So I'm happy to take any questions. Thanks.
Any questions? Just Leah, UCSF. So I would like to know a bit more about the patient effects observed in the game.
Okay, I will tell you, I may have to get my my co-investigator up here to answer the question, but I'll tell you what he told me briefly. And it's basically appears to have a mid-facial hypoplasia, sort of a shortening in the mid-face and a little bit of doming of the skull. Is that a fair summary? Yeah.
And then okay, then you have scans for both male and female, right? And I would be very curious to know if you can see and categorize or tell me what are the main differences between the male and the female.
Okay, so I'll just, before I hand the mic over, just preface saying the confocal data, all of the fish are too young for us to know whether they're male or female, so we can't tell with those. So it's all, yeah, but the micro CT, yeah.
So we just did a manuscript where we went and scanned large amounts of male and female fish, and actually we couldn't find. So in this case, basically, it's normalized for length, which is basically the age of the fish.
Well, Shannon, you're from USC. Do you have any sort of anatomical landmarks that find do some precise measurement?
We are working towards that. So we have been trying to identify a few landmarks, mainly to help register the micro CT and the confocal data. And Osman can tell you a little bit more about that later. And we've also, so I didn't really talk about this, but we've also been trying to use the confocal data and the 3D reconstructions to get volume measurements out for specific bones and doing some comparisons between wild type and mutant based on that. But I agree that it would be good if we could come up with an even better set of landmarks. So we're working on it.
Greg Owens from Icon School of Medicine in Arizona. This may end up being a question for Matthew. With the silver nitrate staining, do you know if that could be applied to mice, like embryonic mice? Consider that?
I considered that. I tried one or two and had some problems with the vulnerability issue, but if that can be broken down by digestion or other mechanical methods that are commonplace in doing poison and red, I think it's very well. It's used on sections already. It's by the Foncasa staining. There's no reason why you can't get rid of it.
What age was the embryo you tried?
It was 16.5.
And you could just remove the skin.
Yeah, but the benefit is somehow you can age if you don't need some kind of digestion.
Okay.
Very nice letter. I was curious with the text. You already mentioned that you're going to be looking at regions to see if there's differences in the life regions. But I was wondering, in the heads, regardless of the knockout, are there certain regions that you're seeing more penetration or more?
So Rich, sorry, are you talking about these fish? That would be very mutation dependent. So I'm not sure. I have to think a little bit. But for example, the chihuahua that I showed, those are always cats because of the dominant cat. That's why human OI patient. And I would say one thing I can comment on, just based on focus data, that although the entire skeleton does end up being eventually somewhat smaller, so like adults are shorter than their wild type siblings, but the skull is more drastically affected than that. So even if we normalize for length of the fish, the skull growth still lags in those. Who else has a question?
Dick Moss, Boston. Could you tell us a little bit more about the craniosynostosis phenotype in late-stage fish? Is the pathway conserved? With FGFR mutants?
So we have several mutants that have sort of a synostosis, and a lot of those affect the extracellular matrix that will come out of the screen. You're looking at the FGFs, there seems to be some dosage balance that's occurring with the FGF1 and FGF3 that we're looking at. We do see extra sutures and failure of the sutures to really sort of be patterned appropriately. Not quite the same as, for example, that mutant that was shown here, which actually has sort of classic synostosis. So right now we're trying to figure that out on a sort of genetic mechanistic level by using some of these techniques as well. But in general, I mean, the dorsal sutures are somewhat conserved for the fluid. And so there may be shifts and balances between specific regulators of that, but the general anatomy of that is relatively good. That would I answer the question?